Hello dear students. In this video, we're going to speak about the factors affecting the rate of photosynthesis. Let's start. The rate of photosynthesis can be measured at the volume of carbon dioxide taken in by a part per unit time, or as the amount of carbohydrate produced per unit time. In laboratory investigations, the rate is commonly estimated as the volume of oxygen released per unit time, which is more easily measured. However, this method does not give an accurate measure of photosynthesis. Some of the oxygen generated by photosynthesis is used by the plant for respiration. Respiration goes on all the time, even when photosynthesis is at its height. So using oxygen liberation as a measure of photosynthesis gives an underestimate of the true rate. We are actually measuring the rate of photosynthesis above a point called the compensation point, defined as the point at which the rate of photosynthesis in a plant is an exact balance with the rate of respiration, so there is no net exchange of carbon dioxide or oxygen. The compensation point is usually related to a particular light intensity or carbon dioxide level. Photosynthesis is affected by many factors, both external and internal. External factors include light intensity, the wavelengths of light, carbon dioxide levels, temperature, wind velocity, and water and mineral supplies. Internal factors include type and concentration of photosynthetic pigments, enzyme and water content, and leaf structure and position. The effect of many of these factors is difficult to determine quantitatively because they interact. They also affect other processes in the plant. For example, the importance of water to photosynthesis cannot be demonstrated easily. Simply depriving a plant of water kills it, but the cause of death may not be connected with photosynthesis. The importance of water can be demonstrated using water labeled with a heavy isotope of oxygen, oxygen 18, and tracing the isotope using an instrument called a mass spectrometer, which can measure the masses of atoms. One batch of chlorella is placed in water in which the oxygen atoms have been replaced by the heavy isotope. Then a second batch of chlorella in unlabeled water is given a supply of carbon dioxide labeled with oxygen 18. Only the first batch of chlorella gives off oxygen labeled with oxygen 18, confirming that the oxygen formed in photosynthesis comes only from water, not from carbon dioxide. Light intensity, carbon dioxide concentration, and temperature are three external factors that are relatively easy to manipulate. Consequently, they have been the focus of many investigations on photosynthesis. The rate of photosynthesis is directly proportional to light intensity. A typical plant responds to changes in light intensity. Very high light intensities may actually damage some plants, reducing their ability to photosynthesize. The light compensation point varies for different plants. Two major groups have been identified: sun plants and shade plants. Sun plants include most temperate trees, such as oak. They photosynthesize best at high light intensities. Shade plants include those of the shrub layer, such as ferns. Their light compensation point is relatively low, but they cannot photosynthesize very efficiently at high light intensities. Consequently, sun plants outcompete shade plants at high light intensities. Carbon dioxide levels. The average carbon dioxide content of the atmosphere is about 0.04%. As long as there is no other factor limiting photosynthesis, an increase in carbon dioxide concentration up to 0.5% usually results in an increase in the rate of photosynthesis. However, concentrations above 0.1% can damage leaves. Therefore, the optimum concentration of carbon dioxide is probably just under 0.1%. In dense, warm, and well-lit vegetation, low levels of carbon dioxide often limit the rate of photosynthesis. Growers of greenhouse tomatoes recognize this and provide a carbon dioxide-enriched atmosphere for their plants. Temperature. Changes in temperature have little effect on the reactions of the light-dependent stage because these are driven by light, not heat. However, the reactions of the Kelvin cycle are catalyzed by enzymes, which, like all enzymes, are sensitive to temperature. The effect of temperature on these reactions is similar to its effects on other enzymes. The optimum temperature varies for each species, but many temperate plants have an optimum temperature between 25 and 30 above zero. Law of limiting factors. So far we have looked at the effects of isolated factors. However, under natural conditions, plants are subjected to many factors simultaneously. The law of limiting factors states that when a physiological process depends on more than one essential factor being favorable, its rate at any given moment is limited by the factor at its least favorable value and by that factor alone. When other factors are kept constant, an improvement in the value of the limiting factor leads to an increase in the rate of the process. Conversely, when the rate of the process does not increase in response to an improvement in an important factor, some other factor is limiting to process. For a process to go at its maximum rate, all factors must be at their optimum level. That is the end of our video session. Next time we're going to speak about photosynthesis in different climates. Thank you for watching.